Sensory drive hypothesis. The divergence of sensory abilities in different species fits well with the sensory drive hypothesis, an explanation for ecological divergence within species, Endler, 1992. Sensory drive describes how communication signals work effectively and how these co-evolve with sensory systems. Basically, speciation by sensory drive is a special case of speciation by natural selection. According to this theory, when populations occupy new habitats with different sensory environments, natural selection favors adaptations that maximize the effectiveness of communication, both within and between species. In other words, the traits showing adaptations are those that improve the ability to send and receive signals in the new environment. The entire process could work in the following way. Individuals of the same species become separated by a geographical barrier, e.g., a river or mountain range, partly because there is competition for resources, such as food or available mates. The new habitat has features that make the transmission of sensory signals more or less difficult than in the previously occupied territory. For example, if birds moved into a new area with denser trees, the light levels would be lower, and it would be more difficult for females to detect the colored plumage on males. Females with better visual acuity, as well as males with brighter feathers, would have a reproductive advantage that was not apparent in the original habitat. This would promote the evolution of both traits in the isolated groups of animals. Other forms of communication, such as aggressive displays or prey-predator interactions, may also be subject to the forces of sensory drive. That is, sensory signals which are transmitted or detected more effectively in a new environment will be favored in terms of reproduction and survival. This topic is discussed in more depth in Chapter 12, Communication. If the sensory drive hypothesis is true, it should be possible to trace the heritability of sensory abilities across generations. Endler and colleagues conducted this experiment in a controlled laboratory setting using populations of guppies, Poacelia reticulate, that were selected for different visual sensitivity. Endler et al., 2001. Guppies are able to see a range of colors across the visual spectrum, although there is individual variability in how well they see each color. This sensitivity can be measured by placing fish in cylindrical tanks with black and white vertical stripes moving around the outside of the tank. See Figure 2.6. Guppies orient to the vertical stripes and maintain their position as they move around the tank. A colored filter is then placed in the light path to illuminate the stripes in red or blue. Fish continue to track the stripes as the light intensity is decreased. At a certain intensity, they stop tracking the stripes, indicating that they can no longer see them. Figure 2.6. Experimental design to test visual sensitivity in guppies. A cylindrical fish tank was surrounded by vertical black and white stripes that moved around the tank at a constant speed. Guppies swam around the perimeter of the tank in line with the movement of the stripes. The stripes were changed to red or blue by placing a colored filter in the light path, and the intensity of the light was slowly decreased. Using this approach, Endler and colleagues separated groups of guppies that were more sensitive to red light from those that were more sensitive to blue light, and then interbred each color-sensitive group. After the 9th generation, fish from each group, including a control group that was crossbred across the red and blue sensitive groups, were tested for color sensitivity. The two color-sensitive groups showed an even greater difference in their visual sensitivity than the original parent groups, indicating that this sensory trait was passed from generation to generation. Endler's experiments were not the first to show heritability of sensory system function, but they provide one of the best examples of how observational field studies can direct experimental research in the lab. Sensory bias. One apparent paradox in the evolution of sensory systems is a phenomenon called sensory bias. Sensory bias refers to situations in which individuals of a species respond with increased vigor to stimuli that are exaggerated versions of naturally occurring stimuli. The phenomenon is often explained as a sensory adaptation that gets co-opted for sexual selection. Figure 2.7 shows a rather comical example of an experiment demonstrating sensory bias. Researchers attached abnormally large feathers of different colors to the heads of male birds, and then assessed female preference for these birds by measuring the time they spent with each decorated male. Burley and Szymanski, 1998, white-crested males were the most attractive to female birds, even though the females had never encountered such elaborate head ornaments on potential mates. Figure 2.7, a male long-tailed finch left and a male zebra finch right, decorated with artificial white crests. During mate choice trials, females preferred to spend time interacting with these birds than with undecorated males or males sporting red or green crests. They look super cute. The paradox of sensory bias is that it cannot provide any evolutionary advantage because the exaggerated trait is not part of either animal's natural environment. The most likely explanation for sensory bias is that it is a byproduct of a sensory preference that had an evolutionary advantage in a different context. This idea, sometimes called sensory exploitation, implies that sensory signals, which were important for one process, have been co-opted by another. In the case of the female finches, their preference for large white headdresses on males may have been borrowed from their nest-building behavior, which uses white feathers, so the females may be hanging out with these males because they have an existing bias for white feathers, not because these feathers indicate anything about male fitness. Sensory exploitation also describes situations in which a trait evolved to capitalize on an existing preference. This two-stage process is revealed by tracing the phylogenetic development, i.e., evolutionary history, of a trait and determining whether a preference for this trait predated its emergence. Basolo, 1990, conducted this analysis by examining the evolution of the tail extension in swordtail fish, Ziphirophis hilari. Swordtail fish are related to the swordless platyfish, Ziophorus maculatus, and their common ancestor also lacked the sword-like extension of the tail. The characteristic fringe on the swordtail, therefore, evolved when the platyfish and swordtail fish diverged. Even though it never existed in their species, female platyfish prefer conspecific males with artificial extensions on their tails, suggesting that this sensory bias for long-tailed males was present in their evolutionary ancestors. When the two species diverged, male swordtail fish were at a reproductive advantage in that this female tendency created a feed-forward loop in evolution. Longer-tailed males were more likely to be selected as mates, producing more males with long tails and more females who prefer them. The latent preference for long tails survived in female platyfish because it was not detrimental to survival or reproduction. It was only revealed when interfering scientists attached extensions to these unsuspecting males. The principles of sensory exploitation may also explain heightened responses to exaggerated versions of natural stimuli, supernormal stimuli. Tinbergen documented this phenomenon in gray-leg geese, answer, when they retrieved and attempted to incubate giant artificial eggs, even while neglecting their own normal-sized eggs that sat nearby, 1951. In other experiments, birds preferred to sit on dummy eggs with enhanced markings, such as more saturated versions of their own egg color. Similarly, stickleback fish, Gasterosteus aculatus, ignore invading conspecifics to attack wooden models of fish with darker red undersides. Ethologists speculated that an animal's natural tendency to respond to the strongest stimulus in the environment leads to abnormal responding to stimuli that have no parallel in the natural world. For example, birds that tend to the largest eggs in their brood as these are likely to produce the healthiest young. If big is better, biggest is best. Since oversized eggs only show up with roving ecologists, there is no evolutionary pressure to have an upper limit on this preference. If this is true, responding to supernormal stimuli can be explained as approximate rules of thumb in a world of normal variation. In summary, a cross-species comparison of sensory systems reveals an enormous range of sensory abilities across the animal kingdom. All animals and humans experience only a limited portion of the sensory world, but these generally reflect the evolutionary history of that species. In other words, sensory system evolution, like other physiological traits, is an adaptation. Although most of this section focused on vision, the same analysis could be applied to any of the other senses in that the auditory, olfactory, and tactile systems of different animals have all evolved within the ecological constraints of particular environments. Development of sensory systems. Evolution provides each organism with a genetic code or set of instructions for building proteins, but how this code is expressed in a fully developed individual varies widely. In some cases, the instructions provided by a genetic code are clear and exact. Build 2 blue eyes. In others, there is a range of possible outcomes for each set of instructions. Build a device that can detect visual signals within a particular range of the light spectrum. Could be accomplished in many ways. The difference in how these instructions are carried out is determined by both physiological and environmental factors. A person may be born with a genotype to produce a tall, lanky body, but malnutrition or excessive consumption of fats early in life would alter the size and shape of their adult build. Sensitive periods. Much of what is known regarding sensory system development comes from the pioneering work of Hubel and Wiesel. These scientists shared the Nobel Prize in Physiology or Medicine, 1981, for their work describing how visual information is processed and organized in the brain. In the course of these investigations, Hubel and Wiesel uncovered a critical role for experience in visual development. It was already known that children who are born with cataracts never fully recover their sight if the cataracts are removed after the age of 3. In sharp contrast, cataracts that develop in adulthood and are removed have little or no impact on vision. Hubel and Wiesel created a parallel scenario in animals. If the eyes of a kitten or monkey were sewn shut during the first few weeks of life, normal vision never developed even if the eyes were later reopened. As little as 1 week of visual deprivation during the first 6 months of a monkey's life was enough to produce this deficit, whereas visual deprivation for weeks or even months in adulthood had no effect. Hubel and Wiesel also showed that cells in the eye which receive light signals respond normally after visual deprivation, whereas neurons in the cortex that receive signals from these cells in the eye do not. This suggested that the connection between the first and final stages in the visual pathway failed to develop under sensory deprivation conditions. The effect was so pronounced that even closure of one eye produced profound structural and functional changes in the cortical neurons that normally respond to visual input. 1977. Hubel and Wiesel concluded that key components of the visual system develop after birth, and that the physiological changes underlying this development depend on visual experience. This period in which experience-dependent changes can have profound and enduring effects on development is called the sensitive period. Knowledge of sensitive periods has improved the prognosis for a number of medical problems, including strabismus. Strabismus is a condition, often present at birth, in which the visual axes of the two eyes are misaligned. This misalignment makes it difficult to look at the same point in space with both eyes, thereby disrupting depth perception and binocular vision. Infants with strabismus have good vision, but as they grow, the images in the two eyes do not converge and the visual system fails to develop properly. Children tend to favor the stronger eye, often losing most of their vision. In the neglected eye, strabismus can be treated surgically, and, in the past, surgery was often delayed until children were old enough to understand the medical procedures. Vision was improved in these cases, but binocular vision often remained impaired. Now that scientists have a greater understanding of sensitive periods and sensory system development, ophthalmologists treat strabismus at a much younger age. If the surgical intervention occurs as soon as the condition is diagnosed, children frequently develop normal binocular vision and depth perception. Sensory deprivation is an extreme example that occurs only rarely in the natural world, but even subtle changes in sensory input during the sensitive period can alter development. For example, if cats are exposed to a particular visual stimulus during the first few weeks of life, e.g., horizontal versus vertical lines, their ability to detect the previously exposed pattern is enhanced in adulthood. This shift in sensitivity occurs because connections between neurons in the visual cortex are rearranged during development, and these cortical neurons become more responsive to the familiar pattern. Blakemore, 1976. Similar findings have been reported in both the auditory and tactile systems of other mammals, in that alterations in early sensory experience modify neuronal connectivity and function in the adult brain. One of the most striking examples of this phenomenon is second language learning. Acquiring a foreign language is remarkably easy as a child and frustratingly difficult as an adult. One problem for late language learners is that they have difficulty distinguishing, and then reproducing, sounds that are not part of their native tongue. Exposure to these sounds during the first few years of life, like visual exposure to lines of different orientations, alters brain development and makes it easier to distinguish foreign sounds in adulthood. The concept of sensitive periods has been extended to other cognitive processes such as categorization, Chapter 9, social competence, Chapter 10, and communication, Chapter 12. Indeed, the idea that early life experiences shape adulthood is the basis of the huge industry built around early childhood education and enrichment. Compensatory plasticity hypothesis. Sensory deprivation reveals a critical role for experience in brain development. If vision or audition or any other sensory input is blocked during development, the functioning of that system may be disrupted in adulthood. Many people believe that these deficits in one sensory system lead to enhanced functioning in other sensory systems. Anecdotally, blind individuals can hear much better than people with normal vision, although this does not prove that the auditory system has compensated for visual deficits when people cannot see. It may simply be the case that those with normal vision have not learned to pay attention to auditory cues because they rely so heavily on vision. Careful laboratory experiments, however, confirmed that blind adults really do have superior auditory abilities, and the effect is confined to those with congenital blindness. Lessard et al., 1998. The question then arose whether people who are born blind have better hearing at birth, or whether this developed because visual input was restricted. Theoretically, this issue could be resolved by testing audition in blind and sighted babies, but the experiment would be difficult to set up and may take years to complete. Researchers would need to identify and test blind newborns with no other medical condition, and then match these to babies with normal vision. In contrast, an animal experiment to test this hypothesis can be designed with relative ease. Chapman et al., 2010, conducted this work by raising guppies under different light intensities and then testing how they responded to visual or chemosensory, i.e. olfactory, cues. The prediction was that fish raised in environments with minimal visual input would develop improved olfaction. This is exactly what happened. When tested for their ability to find food, guppies reared at high light intensities responded better to visual cues, whereas those reared under low light intensity responded better to olfactory cues. Since the guppies were randomly assigned to each lighting condition at birth, the group differences must be due to their environmental experience early in life. These results fit the compensatory plasticity hypothesis, which states that a loss or deficit in one sense leads to a heightened capacity in another. Compensatory plasticity appears to be a developmental process in that altered sensitivity to visual and olfactory cues only occurs in early life. Older guppies were unable to respond to similar changes in their sensory environment. That is, fish reared under low light conditions did not show improved visual responses when they were tested under high light as adults. It appeared that once the olfactory system became dominant in these animals, it could not be reversed. The advantage of compensatory plasticity is that it allows animals to adapt, within their lifetime, to new sensory environments. Natural selection provides a mechanism for adaptation across generations, but this process is random and may be too slow to ensure survival. This is particularly true in aquatic systems that are easily altered by weather and human activity, dams, irrigation, pollution, etc. Compensatory plasticity may be an adaptive mechanism to deal with these rapidly changing sensory conditions. How the process occurs is still not clear. The most likely explanation is that areas of the brain that were genetically programmed to respond to one sense, e.g. vision, are taken over by another sense, e.g. olfaction, when sensory input is reduced.